Morning everyone. This is JIAS Academy Ramburam, Chennai. We are giving online and offline courses. Helpline is given here. CSIR NET, Life Science, Unit 8, Part 3. This class is about Tetrad analysis in gene mapping. Tetrad analysis. Paired chromosome, that is homologous chromosomes, is called a tetrad since they are having 4 chromatids. You can see here these two are called as homologous chromosomes. They come together during the cell division, and each chromosome divides into 2 chromatids. So 4 chromatids are called tetrad. Tetrad analysis is a special mapping technique, can be used to map the genes of haploid eukaryotic organisms which are the product of single meiosis event. Ordered tetrad. What is ordered tetrad in the sense? It is used to calculate the distance between gene and centromere. See, what is ordered tetrad? Ordered tetrad is used to calculate the distance between gene and centromere. See, let us take this is the chromosome and this is the chromatid. Chromosome, at the time of cell division, it divides into 2 chromatids. So this is the centromere and here the gene. Let us name it A. So this is the gene and this is the centromere. So ordered tetrad is used to calculate the distance between the centromere and the gene. Okay. Relative proportion of tetrad is used to calculate the map distance rather than the progeny. Then for this genetic studies, a plant, that is, it is not plant actually, a fungi, Neurospora grassa, it's a fungi which is commonly called as orange bread mold. You can see here, see this is the fungi. Okay, so orange bread mold it is called as Neurospora grassa. Its name is Neurospora grassa. This is botanical name, and commonly it is called as orange bread mold, which revealed that the crossing over occurs after the duplication of the chromosome. The crossing over occurs after the duplication of the chromosome. Okay, so the Neurospora produces ordered tetrad that we can see here. See, this is called as ascus. This is called as ascus, and the nucleus are arranged here in a linear manner. So this is called as ordered tetrad. Okay, tetrad in the sense only four. See, if you imagine this is the ascus, and tetrad in the sense only 4. First meiotic division, result of this one, and second division, each one mitotic division takes place and it produces 4 more. So you can see here 8 actually. Okay, these are called as ascospores, and this organ or this part of this fungi is called as ascus. One interesting question is why Neurospora grassa was taken for the study. There are a few reasons that why the Neurospora grassa is taken into genetic studies. Let us see one by one. The meiosis occurs in diploid nucleus formed after the fusion of the nuclei from 2 distinct strains. You can see this one in this picture. See here, this is one strain, that is type A strain, and this is type B strain. These two, there is a fusion of conidia from A and B. They fuse to form the zygote, and from the zygote, meiosis cell division occur, and these divides into 2. Okay, divides into 2, that is this, the B as well as A. Let us take this is B and this is A. Now this B divides into 2 and this A divides into 2, and this is called as tetrad. This is called as tetrad. This is the first meiotic division is the reduction division. Second meiotic division, which gives rise a tetrad, and after this mitotic division takes place and it gives an octad. Octad in the sense 8 spores are present here. Okay, it is 8. So here the mitotic division takes place and produces this octad. Okay, and then these spores are released outside. So this is what the first meiotic division. First meiotic division is the reduction division. Second meiotic division is similar to the mitosis. So this forms 4. Okay, 4 spore. That stage is called as tetrad, and after this formation there is a mitotic division takes place and it forms the 8 ascospores. Okay, ascospores. So that stage is called as octad. Then after mature of this ascus, this will be ruptured and released outside. Okay, so this is what I have shown here. So this ascus is in octad stage. Okay, so it is very easy to study. So this fungi is selected for the genetic studies. Neurospora grassa, and the haploid product of meiosis are called ascospores. I told you these are the ascospores, and they contain in a linear order in a tube-like structure. This structure is called as ascus. Okay, and this ascospores are germinate to produce the haploid mycelium. This you can see here. This cycle is called a sexual cycle, and this is called as asexual cycle. That is, fusion of gonadia form the ascus. So that is called as sexual cycle. And here what happened? These spores, these ascus, they form the vegetative mycelium. This may take asexual cycle or a sexual cycle, depend upon the favorable environment. See, if you take the asexual cycle, the gonadium, this germinate and they produce a vegetative mycelium. Then this vegetative mycelium forms, after maturation, it gives the gonadia. You can see here. Okay, if this gonadia falls under the favorable condition, then from this gonadium the new mycelium develops. Okay, this spore germinate and it forms a vegetative mycelium. And in the sexual cycle, if you see the mycelium, the 2 strains of mycelium, and from that the gonadium, they fuse together, that I have shown here. These gonadias fuse together and they form the zygote, and from the zygote these tetrad and octad will be formed, and then these spores, ascus spores, are released outside. Okay, so this is very easy to study. So the genetical studies are very easy in this fungi. That fungi is named as Neurospora grassa. The ascospores germinate to produce a haploid mycelium. So the allele, irrespective to dominant or recessive, can be determined very easily in this haploid phase. So why this fungi is selected? Neurospora grassa can be grown very easily on synthetic medium. So this is also one of the reason why the scientists selected this fungi. Then haploid phase can be maintained asexually. The order of the 4 spores within the ascus reflects exactly the orientation of the 4 chromatids of each tetrad at the metaphase at meiosis 1. The spores can be isolated in the same order as they are in ascus. Okay, so for these reasons, For these reasons, the scientists selected Neurospora grassa for genetic studies. And I have shown this asexual cycle of this En grassa and sexual cycle of Neurospora grassa. I have shown you. This is the ascus. From this ascus only, the octed ascus will be formed, and it releases the ascospores. These ascospores fall into the fallen down, and when it reaches when it gets a favorable condition, then it forms or grow into a vegetative mycelium, and that select according to the condition, environmental condition, it gone under sexual cycle or asexual cycle. Okay, commonly it is called as, we have seen this one, commonly it is called as orange bread mold. The botanical name is Neurospora grassa. The next one is mapping of gene by Tetrad analysis. Mapping of gene by Tetrad analysis. So it is used to map on the locus of the relation to the centromere. This is the centromere. In Neurospora grassa, meiosis is followed by mitotic division. Meiosis is followed by mitotic division, in the sense meiosis is a reduction division. Okay, reduction division in the sense 2n number of chromosome is reduced to n number of chromosome. That is followed by the mitotic division. Okay, mitotic division resulting to 8 ascospores arranged in 4 pairs in the ascus. Each pair representing one of the product of the meiosis. For example, if a mating of A is crossed with the N grassa, resulting the zygote will be, A is crossed with small a, in the sense the zygote will be AA. Okay, so it possesses such allele in the chromosome. And in the first case, if you take, when there is no crossing over, so this is the gonadea, type A gonadea, this is type B gonadea. So it is giving rise the zygote, there is no crossing over. So all the tetrad in the sense, this possesses type B. This is type B chromosome is present inside, and here type A chromosome is here. And this gone through a mitotic division and gives rise to 4 different ascospores. All these 4 different ascospores possess only A type of chromosome. All these ascospores produce, or in the ascospores there is B type of chromosome. Okay, so there is no crossing over. A pure offspring or progeny is produced from the ascospores. Okay, this is ascus. From the ascus, 8 different ascospores are produced. These 8, among these 8, 4 possess the A chromosome, A allele chromosome, and the other 4 possess B type of genes. These comes from that is same as that of the parents. 4 are same as that of the type A parent, and 4 are type B parent. Okay, this is called as, so when there is no crossing over between the mating type of locus and its centromere, this type of segregation is called as first division segregation. That is FDS. FDS means first division segregation. Okay, first division segregation. This type of ascospores are formed. Half of the ascospore possess A type of chromosome, A type of gene, and half of the ascospore possess B type. Let us see the second one. In the second case, if crossing over occurs between the gene and its centromere, then A and A allele do not get separated until the second meiotic division. until the second meiotic division, this won't get separate. Okay, A and A won't separate. See, you can see here, this is crossing over occurs between the gene and its centromere. Here you can see very clearly, this part you can see very clearly. Actually, this blue is A, okay, which possesses the A gene, in the sense this red possesses the small a gene. So here the crossing over occurs. So this part is shared to this chromosome, and this part shared to this chromosome. Okay, so if you see the gene, this chromosome possesses A, capital A and small a, and this also possesses capital A and small a. That is, this capital A comes from this chromosome, and this a small a comes from this chromosome. Okay, so this is ordered tetrad. Ordered tetrad in the sense the the alleles are arranged in a particular order in the chromatid. So it is called as ordered tetrad, and each chromatid is going to give a ascospore. So it is called as ordered tetrad. It is called as, and this division is called as second division segregation. Okay, SDS, FDS and SDS. FDS in the sense first division segregation, SDS in the sense second division segregation. Okay, see, this may be in the form of, if the crossing over occurs, this may be in the form of this pattern, or in this pattern, or in this pattern. Okay, next one, calculating map distance. How to calculate the map distance? Okay, so to convert the tetrad data to crossover or recombination data percentage of second division segregation assay is divided by 2. That is, map distance is equal to half into number of SDS assay divided by total number of assay into 100. Okay, this is the formula for calculating the map distance. Okay, this is very, very important. So make a note. This formula is very, very important, and they'll ask what is SDS and what is FDS in the sense first division segregation, second division segregation, and the formula map distance in the sense half into number of SDS assay divided by total number of assay into 100. Next one, if you see tetrad analysis to map two linked genes. In some cases, the product of meiosis do not retained in a linear manner. Inside the single structure get mixed. These type of tetrads are known as unordered tetrad. Okay, see, ordered tetrad we have seen there. An unordered tetrad, how it is in the sense the product of meiosis do not retained in linear manner inside the single structure, and it get mixed. So this type of mixed is called as unordered tetrad. Okay, and there are 3 tetrad types: T, PD and NPD. And what are they? We are going to see now. These are produced when 2 genes are present in the same chromosome. See, previously we have seen a single chromosome, but here if there are 2 genes in the same chromosome, this may happens. Okay, so how? Let us see now. Condition number 1, when there is no crossing over occurs, or 2 strands double crossing over occurs between 2 loci, then PD are produced. Okay, PD are produced. What is that? When there is no crossing over or 2 strands. Okay, double crossing over. What is this? This is called as 2 strand crossing over. 2 strand. You can see here, these are the 2 homologous chromosomes. They come together, and this possesses 2 chromatid, this possesses 2 chromatid. So one chromatid from this side and one chromatid from that side, they fuse. Okay, they cross. So this is called as 2 strand double crossover occurs between 2 loci. You can see this is one loci and this is another loci. Okay, so there is 2 crossing over occurs between 2 loci. Then this is called as PD. Okay, PD in the sense parental ditype. PD are produced. Okay, PD are produced in the sense parental die type are produced. Okay, so you can see here 2 points, the crossing over occurs here. Okay, next one, condition 2, a fourth strand double crossover between two genes result in two kinds of spores that do not resemble the parent. Here they resemble the parent, but here they do not resemble the parent. So why it is called as NPD, that is non-parental ditype. Non-parental ditype. What is that? Four strand crossing over occurs. That is this type. Four strand two genes, the crossing over is between two genes. Okay, so this is called as non-parental ditype. Okay, ditype, non-parental ditype, this is okay. And condition number 3, two strand single crossover or three strand double crossover between two genes result in the formation of both parental and non-parental or recombinant TT. Okay, that is Tetrad type. You can see here 3. Okay, sometimes 2 may cross or sometimes 3 may cross, and they form half like parents and half is non like parent. So this is called as Tetrad type. This is called as Tetrad type or recombinant TT. Okay, this is called as recombinant TT. So we have seen 3 types. One is parental PD, okay, parental ditype, and another one is NPD, non-parental ditype, and another one is three standard. This may be in the form of crossing over in the form of this type or in the form of this type. Okay, these two may occur, and half is like parent, half is not like parent. Here these two in 3 strand. Okay, 3 strand, these two, and this is 4 strand, and this is 2-point crossing over between 2 strands. Okay, so condition number 1, condition number 2, condition number 3 we have seen here. Okay, this is 2 linked genes. Tetrad analysis to map 2 linked genes. Okay, now in Tetrad analysis we analyze all type of tetrad rather than the progeny. The basic formula for this is half T plus NPD divided by total tetrads into 100. This is the formula for distance between two gene. Okay, distance between two gene can be calculated by using this formula. Okay, so thank you so much for this class. Okay, share this class to your friends, those who are preparing for CSIR NET. Okay, the email ID and our website address is given here, so you can contact our office. Helpline is also here. Okay, so thank you so much.